A very warm welcome to all the listeners of the MedSynapse podcast series on gastroenterology. Gastroenterology podcast focuses on the latest clinical and research developments in the field of gastroenterology. Today's podcast episode focuses on the trial, titled Effectiveness of Severe Acute Respiratory Syndrome Coronavirus 2 Vaccination, in a Veterans Affairs cohort of patients with inflammatory bowel disease with diverse exposure to immunosuppressive medications, published recently in the Journal of Gastroenterology. The severe acute respiratory syndrome coronavirus 2 pandemic has caused a great impact on the general population and their health as well. To curb the ongoing pandemic caused by severe acute respiratory syndrome coronavirus 2 infection, vaccine development has been undertaken and has been in use widely. Two vaccines that are in wide clinical use are BNT162b2 messenger RNA coronavirus vaccine from Pfizer, and the messenger RNA 1273 severe acute respiratory syndrome coronavirus 2 vaccine from Moderna. Inflammatory bowel disease, that is IBD, is one of the diseases that can lead to severe infection. Immunosuppressive medications play an important role in reducing the risk of IBD. Thus, this trial mainly focused on the effectiveness of severe acute respiratory syndrome coronavirus 2 vaccination in the IBD population, and the potential impact of immunosuppressive medications. This was a retrospective cohort study of patients in the Veterans Health Administration with IBD diagnosed before December 18, 2020. The cohort comprised 14,697 patients, 7,321 of whom received at least one vaccine dose. IBD medication exposures included mesalamine, thiopurines, anti-tumor necrosis factor biologic agents, vedolizumab, ustekinumab, tofacitinib, methotrexate, and corticosteroid use. Patients who were 18 years of age and who had not previously been diagnosed with severe acute respiratory syndrome coronavirus 2 infection were included in this study. It was found that the median age was 68 years, wherein 92.2% were men. Those who were not vaccinated faced higher chances of getting the infection. Full vaccination status was associated with a 69% reduced hazard of infection relative to an unvaccinated status. There were no significant differences associated with key immunosuppressive medication classes. Results demonstrated that patients with IBD did not inherently have an increased risk of developing severe acute respiratory syndrome coronavirus 2 infection or associated complications of COVID-19. The risk of severe acute respiratory syndrome coronavirus 2 infection was not reduced for patients who received a single dose of vaccine. In addition, study demonstrated that COVID infection vaccine effectiveness was similar in patients with IBD regardless of the particular immunosuppressive agents being used. One major strength of this study was the use of a national cohort of IBD patients monitored in the Veterans Health Administration system, as it helps in keeping the record of all COVID infection diagnoses and vaccination status. One major limitation of this study was that the patients were not screened for coronavirus infection in the Veterans Health Administration, but rather they were simply tested when symptomatic or for preventative measures. Hence, the patient population could be biased towards symptomatic patients with COVID-19. Based on this trial, we can conclude that full vaccination but not partial vaccination was significantly associated with reduced coronavirus infection. It has 80.4% effectiveness in a broad IBD cohort with diverse exposure to immunosuppressive medications. With this, we conclude today's gastroenterology podcast. Thank you for listening.